The first speaker is Luke Tait. He is a research associate at the Cardiff University Brain Research Center, and he's going to talk about his work on optimal source reconstruction of resting MEG of the human brain.
Great, thank you. So I'll just share my screen with you. Great, so hopefully you should be able to see my screen. Yeah, perfect. So as mentioned, I'm going to talk to you about some work I've been doing looking at source reconstructing resting state MEG data. So just to give you a quick idea, the idea of source reconstruction is given some MEG or EEG activity recorded outside of the scalp. We want to estimate the brain activity that generates this data. Now, in the literature, there are many, many different methods for source reconstructing EEG or MEG data. So we wanted to ask, what is the best way to do this? Specifically for resting state MEG or single trial data, which is distributed across the cortex, low signal to noise ratio, compared to, for example, evoked potentials. So to address this problem, we took 11 participants with 30 seconds epoch of pre-processed, artifact-free resting state MEG. So we used a 274-channel CTS scanner for each participant. And the forward model, we used a single shell head model derived from a T1 weighted MRI. We studied 6 commonly used inverse algorithms. So there were 2 beamformers, the LCMV and its depth weighted counterpart, that here we've called weighted LCMV, and then 4 least squares minimum norm type estimates that are common in the literature. So in order to compare the algorithms and quantify which are the best algorithms, or which perform well in different aspects of MEG data, we wanted to look at spatial and temporal quantifications. So for the temporal aspect of the data, we wanted to say, can the algorithm explain the temporal fluctuations due to brain activity? So we took our resting state MEG, we left out a subset of electrodes. So here I've only shown 1. In fact, it was between 10 and 11, and we did 27-fold cross-validation. So we left out a subset of electrodes, and we source reconstructed the remaining electrodes. Then, given our source solution, we forward mapped back to these left out test sensors. And we calculated the temporal variance explained, so the correlation squared between the predicted activity at those sensors and the real activity that was recorded at those sensors. So there's a slight downside to this method in that MEG data resting state MEG data has low signal to noise ratio. So explaining all of the variance means that you're also explaining the noise in the data. So in order to try and penalize noise and account for this bias, what we did was we also calculated this metric in empty room data, where there is no brain activity at all. And then we divided by the variance explained in that. So now we've got a ratio of with a brain versus without a brain, how much variance do we explain? So that is to explain temporal fluctuations in the data. What about the spatial aspects? To do this, we use something called the resolution matrix. Now, I won't go into the details here, but this is a theoretical relationship between the estimated source activity and the true source activity that we can't know. And from this matrix, we can get something called the point spread function. But that says, if I've got activity at a dipole, here marked with a diamond, where does the estimated activity from this dipole, where do we estimate this activity is? And so we can look at something called the localization error, which is the distance between the dipole and its peak of the estimated activity. Or we can look at the spatial extent of this. How far does it spread out? So here we've called it spatial extent of point spread. So this says, how far does leakage come from the dipole? We can also look at something called the crosstalk function that basically says, given an estimate at a dipole, how far away is activity influencing this estimate? So we can look at the spatial extent of the crosstalk function to calculate how far away is leakage into the dipole. So we've got out from the dipole and into the dipole. So these are our metrics to look at the spatial aspects and quantify how good spatial reconstruction is. And finally, we know that resting data is low signal to noise ratio. We can't average over many trials to reduce noise. So we wanted to see, is the algorithm strongly biased by noise? We can't reduce the amount of noise very easily, but we can increase it. So to quantify this, what we did is we first of all source reconstructed our resting state MEG, and then we added some sensor noise and source reconstructed this. And then we performed a spatiotemporal correlation between these two solutions to see how much the solution changes with the addition of some noise. So first of all, we said the temporal fluctuations, the R-squared analysis. And what we found was that the beamformers greatly outperformed the least squares minimum norm type estimates. And this was actually primarily due to the fact that they explained very, very little of the empty room data. They were, whereas the others slightly overfit to the noise. How far away was the activity misplaced, the localization error? Well, we found that the LCMV beamformer had the highest localization error, whereas, as theoretically expected, S Loretta and E Loretta have 0 localization errors. The two spatial extent of leakage measures gave very similar results, actually. So I'll present these together just as a general measure of leakage. As you can see, the beamformers performed worst of the group. They had the highest leakage, whereas the weighted minimum norm estimate had the lowest leakage. And finally, with the noise analysis, with the adding the noise, so on the x-axis here, we've got the amount, the variance of the noise as a ratio of the variance of the data. And you can see for different amounts of noise, the MNE and weighted MNE solutions, the correlation drops a lot faster than the other solutions. And this just sort of quantifies it as the sort of the slope of this drop off. So you can see MNE and weighted MNE drop off a lot quicker. So that's a lot of information in a short amount of time. So I thought it'd be handy if I summarize this all in a big table, summarize these results and give some conclusions. So first observation was that the depth weighted metrics, the weighted LCMV and the weighted MNE, always performed better than or equal to the unweighted counterparts, LCMV or MNE, in all of the metrics that we looked at. So based on our results, we should prefer the depth weighted metrics. This is kind of expected. The depth weighted is supposed to improve the result. So I've left out the unweighted metrics just to clean up the table a little bit here and just looked at the remaining 4. So I would say if the data, we know it's particularly noisy, we know it's noisier than, noisier than the typical analysis, I would recommend using a beamformer. They explain very little of the empty room data and were very robust to noise. So the extra noise is less likely to influence your results. However, this does come at the cost of having high localization error and high leakage. If you prefer to use a least squared minimum norm method over beamforming for whatever reason, and the data is still very noisy, you've got your noisy data, then S Loretta was the way to go. It had, out of all the metrics actually, the highest robustness to the noise, but it did drop in the variance explained. So again, you win some, you lose some there. If having high resolution is your top priority, if you really want to minimize leakage and are happy to take in the localization error, the less robustness to noise, bias to noise, a loss of variance explained, then by all means use the weighted minimum norm estimate, but it comes with many downsides. And finally, if you don't think the data is particularly noisy, if you think it's just fairly standard resting state data, if you don't have particularly strong requirements that it has to be minimize leakage as much as possible, I would say of the group, E Loretta performed generally the most well across the board. It was very rarely the best, but it performed reasonably well in most metrics. So I think E Loretta is a strong choice here. Now, in resting state analysis, typically, actually, we would parcellate the data into regions of interest as opposed to just perform the analysis on the level of voxels. So I won't go into details here just because of time constraints, but if you look at the preprint given in the chat, we reduced the human connectome project's multimodal parcellation down to 230 ROIs specifically targeted towards use with MEG. So this atlas is freely available for download at my GitHub page given down here. We also validated this atlas in a larger sample of 31 participants with 2 scans per participant. So this isn't just the 11 participants that we used in this data, in this analysis. So we modified our metrics slightly to be more suitable for ROI level analysis and re-evaluated these metrics. So I'll just show you again another summary table here. So the first thing that I really noticed was the fractional localization error. So this was, instead of just saying what is the distance between the dipole and the peak of the estimated activity, I said what fraction of dipoles have a peak of activity outside of their ROI. It doesn't matter if the peak of the activity is inside the ROI but a few millimeters away, but it does matter if we're putting it in the wrong ROI. And both weighted LCMV and weighted MNE had over 50%, in fact, over 65% really, of dipoles misplaced into the wrong ROI. So I should say here, this is a very high resolution atlas. If we use, say, the AAL atlas, which only has 78 regions, this might be less of a problem. But for this high resolution atlas, it really means that this is quite a problem using these metrics. So given that we don't want to use either of these metrics for this atlas, we want to use either E-Loretta or S-Loretta. And actually, if we just look objectively at the solutions, E-Loretta outperformed S-Loretta. There's not much between them, but E-Loretta outperformed them both slightly. So I would recommend the use of E-Loretta for high resolution atlases. So that's all my slides. Are there any questions?
Thank you. And it was perfectly on time. Let's see if we have questions from the audience. I saw someone had raised their hand in the audience, but I don't see a question in the Q&A box. Did you have a question that you wanted to type in?
Yes, please send your question in the Q&A section, and we will address it live. If you would like to, after you put it through the Q&A, we can bring you forward to discuss the question further in depth with the speakers as well. Oh yeah, Marco, you can go with the question as well. You can unmute and ask your question.
Okay. So first of all, thank you. Just a great presentation.
Thank you.
I was wondering, how do you think this would compare with task data? Is there any reason for it not to be the same type of results?
So, yeah, I mean, the main reason that I don't want to strongly say that this would also apply to task data is because some of the measures rely strongly on the covariance matrix, so particularly the beamformers rely on the covariance matrix of the data. And if you are averaging over many trials, this is going to greatly change the covariance matrix. Also, these are all regularized solutions, so changing the SNR might change the results as well. You can absolutely apply these analysis methods to the resting state data. And at the GitHub page that I showed on my Atlas slide, at this GitHub page, I've also provided codes to do all these analysis on task-related data. So it's absolutely something you can do, but I don't want to say now that it would work for task data just because we haven't done it.
Okay. So just another question. The decrease on the variance when you move to the Atlas version on the beamformer method, was that expected?
So the reason for this is in the actual paper, we show the R-squared and the empty room data separately. In the Atlas version, what we found was that all of the metrics throughout only explained like 10 to the -4 variance once we parcellated. So I suspect we use the first principal component of all dipoles within an ROI to parcellate. I suspect by rejecting all the other principal components, we're sort of rejecting noise. So in this case, we actually relied just on the variance explained, which was lower for the beamformers in the original solution. It's just that in the voxel-wise data, the least squares minimum norm explain noise as well, whereas that doesn't happen in the parcellated solutions.
Okay, maybe just one last question. How do you think this will generalize to EEG data? Would it be the same? Do you think it?
I think so. There has been some people looking at this with the resolution metrics. It's quite dependent on the EEG, the density of the EEG. I think if we had very, if you had very high density EEG, the results would be fairly consistent. Of course, you've got the difference in the radial versus tangential, which means that, so I haven't included here, but we did plot, because each of these metrics are per dipole, what we showed here is averaged over the whole brain. We did show plots, and that I think the spatial distribution would be different, but I think it's largely.
Sincere apologies for interruption, because we are running more than a minute over time. We might hit the spot where our webinar will be just ended and we'll be kicked out. So yeah, let's move on.
No problem.
Keep the Q&A going in the chat. Just keep talking about it. This is interesting. I'm learning stuff from this as well.
Thank you.
Let's transition to the next speaker. It's